Welcome, welcome, everyone. Janet, you've been here for 2 seconds. Your hand is up already. You don't even know the rules yet. You can't put your hand up.
I allowed Janet to unmute if she wanted to say something.
Yeah, go for it.
Oh, okay.
I can't hear you.
You can't? Okay. I just wanted to make sure I was not muted. For some reason, I can't see my icon.
Yeah, I'll talk about that in a minute, but because of the format of this meeting, actually, Andrea has control over everyone's mute and unmute capabilities.
Okay, good. I just didn't want to make any background noises.
Yeah.
Thank you.
You're welcome. All right, why don't we get started while folks are still filtering in. Thank you. First of all, thank you all for coming. Thank you all for getting up out of your couch. If you're up out of your couch, I don't know if you are or not, but you're here and I appreciate it. And we're gonna have a great and informative evening. So I appreciate you all being here. I just wanna start with a few, I guess, not ground rules, but just some description about how the system is gonna work. So because of the type of meeting that this is, you will not be able to use your own microphone, to turn your own microphone on and off during the talk. There, you should notice at the bottom of the controls, there's a Q&A box that is not usually there when you're using Zoom. So you can use that if you want and type questions in. You can still use the chat, of course, but if you have a specific question, please type it in the Q&A box. You can also raise your hand and Andrea will monitor that and can interrupt, and then you can make a comment or ask a question if you want during the talk. But I think the main idea is to restrict most questions, unless you need a clarifying question, to the end of the talk during the Q&A session. Andrea, did I miss anything, or is that? Oh, the other thing is we are recording this. So if you don't want to be on video, then don't turn your video on when you ask a question or during the Q&A session at the end. If you didn't do your hair today. Okay.
Yeah, KJ, I think that covered it. So thank you for
Sweet.
checking.
You're welcome. All right. So I now get the pleasure of introducing Meghan Bucher. Meghan Bucher is currently in New York, so thank you for being here with the 3-hour time change. Meghan received her Bachelor's of Science in Neuroscience and a PhD in Neuroscience from the University of Pittsburgh. Her dissertation research, which is what she's going to talk about today, which she told me earlier, has just been published, so congratulations. Investigated endogenous dopamine neurotoxicity and was conducted under the mentorship of Dr. Teresa Hastings. Currently, Dr. Bucher lives in New York with her 2 cats, working as a postdoctoral research fellow in the Mailman School of Public Health at Columbia University, studying environmental determinants of dopamine neuron toxicity under the mentorship of Dr. Gary Miller. Today's talk is titled Acquired Dysregulation of Dopamine Homeostasis Reproduces Features of Parkinson's Disease. So virtual clap, welcome and thank you for being here. And I'm going to turn it over to Dr. Bucher.
Great, well, thank you so much for that introduction. Let me share my slides. Okay. So yeah, first I'd like to thank KJ for that introduction, as well as Dr. Finley for the invitation to speak with everyone tonight, and Andrea, who has helped me coordinate everything with this virtual seminar tonight. As KJ mentioned, I'm currently a postdoctoral research fellow at Columbia University, but today I will be presenting some of the work I performed at the University of Pittsburgh while obtaining my PhD. And the title of my talk is coming from the publication of this work. So acquired dysregulation of dopamine homeostasis reproduces features of Parkinson's disease. This is available online through the journal NPJ Parkinson's disease, if anyone is interested in looking at that. And this title is pretty jargony, but as I go through my talk, I will be dissecting the segments of this. So what dopamine homeostasis is, what it means to acquire dysregulation of dopamine homeostasis, and how this informs us about the pathogenesis of Parkinson's disease. And before I get started, I'll just like to say, if you have a question, please feel free to interrupt me during the talk, and I'd be happy to answer or offer any clarification. So I'll start with an introduction to Parkinson's disease. Parkinson's is the second most commonly occurring neurodegenerative disease after Alzheimer's disease. It affects about 1 in every 100 people over the age of 60. Parkinson's is a movement disorder, and it's characterized by debilitating motor deficits. These include rigidity, postural instability, and resting tremor. And it's known that the motor deficits of Parkinson's disease are the result of dopaminergic degeneration. So this is a degeneration of dopamine neurons in the nigrostriatal pathway. These neurons have their cell bodies in the midbrain in an area called the substantia nigra, and they project and terminate in the caudate and the putamen, and collectively we refer to this area as the striatum. And so as these neurons degenerate, there's a deficiency in dopaminergic transmission here in the striatum, and that's what's causing the motor deficits. There are symptomatic relief available for patients with Parkinson's disease. This is targeting that dopamine deficiency to relieve the motor symptoms, but there are currently no cures or disease-modifying therapies for Parkinson's disease. And by disease-modifying therapy, this would be an intervention that would slow or halt the progression of Parkinson's disease. A recent study estimated that the economic burden of Parkinson's disease is about $52 billion a year. And with our aging population, it's estimated that the prevalence of Parkinson's disease is going to increase from around 930,000 people in the United States in 2020 to around 1.2 million people in the United States by 2030. And so with this debilitating nature of the symptoms, the increasing prevalence and lack of disease-modifying therapeutic interventions, it's essential that we continue to study what causes Parkinson's disease, but also to test novel therapeutic interventions. The pathology of Parkinson's disease is well characterized. So as I mentioned on the previous slide, the dopamine neurons that degenerate in Parkinson's disease have their cell body in the substantia nigra. And this region of the brain gets its name from this black substance. So black substance, substantia nigra, which is neuromelanin. Neuromelanin is a polymer of oxidized dopamine. And so what you see in postmortem tissue from a healthy brain is this really apparent black substance here, which is showing that the dopamine neurons are there. However, in postmortem tissue from a Parkinson's patient, there is a loss of the black substance indicative of degeneration of the cell bodies. So these are the cell bodies, and if you look at the terminals in the striatum, you can visualize the dopaminergic terminals by staining for tyrosine hydroxylase. Tyrosine hydroxylase is the rate-limiting enzyme in the production of dopamine. So performing an immunostain, you can see that in a controlled brain, there's this nice dark, dense staining in the striatum showing you where all the dopaminergic terminals are. But with Parkinson's disease, there's a loss of this staining that is progressive with disease progression. And so this is showing that the terminals are degenerating. Parkinson's disease is a proteinopathy, and it has protein aggregates called Lewy bodies. The major component of Lewy bodies is alpha-synuclein, which aggregates and becomes insoluble. And it's also known that neuroinflammation plays a large role in disease process. So this is interactions between neurons and the supporting immune cells of the brain, glial cells. And while the pathology of Parkinson's disease is well characterized, there's still a lot of work to be done to understand what causes Parkinson's disease. So there's no singular cause of Parkinson's disease, and it's known that both genetic and environmental factors are involved in the pathogenesis. The heritability of Parkinson's disease is estimated to be about 0.27 or 27%. So this means that the majority of disease risk cannot be explained by genetics. Analysis has identified some genes involved in the development of Parkinson's disease. So as I mentioned, with Lewy bodies, the major component being alpha-synuclein, we know that mutations or gene variants in alpha-synuclein can increase the risk of getting Parkinson's disease. Monogenic inherited forms of Parkinson's disease only account for about 10% of cases. So these genetic variants identified, we mostly classify them as predisposition genes. So if you have the variant, you're more likely to get Parkinson's disease. And it's also known that environmental factors such as pesticides or chemical agents can be linked to the development of Parkinson's disease. And so just like purely genetic forms of Parkinson's disease being rare, Parkinson's disease resulting strictly from environmental exposure is also very rare. So it's believed that the majority of Parkinson's cases result from interactions between these predisposition genes, as well as a lifetime accumulation of environmental exposures. And this is also paired with normal deficits that occur as we go through the aging process. And so while these factors involved in the development of the disease have very diverse functions or very diverse targets in the cell, they converge on common pathogenic mechanisms. And so these include mitochondrial dysfunction, oxidative stress, and protein accumulation. This is not a comprehensive list of all of the pathogenic mechanisms involved in Parkinson's disease, but these are the features that I will be focusing on in my talk. So these genetic environmental factors are ubiquitously experienced throughout the body and the brain, but not all neurons are affected by these. Not all neurons degenerate because of this. So it's important to understand why only specific neuronal populations undergo degeneration. And while other neuronal populations are affected in Parkinson's disease, those dopaminergic neurons in the nigrostriatal pathway seem to have enhanced susceptibility to degeneration. They degenerate faster and to a greater extent than other neuronal populations. And so trying to understand what makes these dopamine neurons more vulnerable to degeneration than other neurons, one hypothesis is that it's actually dopamine itself that is contributing to the vulnerability. So as we've established, the ability to control movement depends on dopaminergic transmission from the nigrostriatal dopamine neurons. And as we see in Parkinson's disease, the loss of dopaminergic transmission results in motor deficits. So it's essential for dopamine to undergo proper synthesis, packaging, release, reuptake, and metabolism. So these are what I consider to be dopamine homeostasis in order for dopamine to be released and movement to be functional. So the two features of dopamine homeostasis that I will be focusing on in my talk are going to be packaging and metabolism. And to start with the packaging of dopamine, so in dopamine neurons on the synaptic vesicle, there is this protein, a V-type ATPase. And what this protein does is it uses ATP to pump hydrogen ions, protons, into the interior of the synaptic vesicle, and this is against their concentration gradient. This accumulates hydrogen ions inside the synaptic vesicle, and this is important for two reasons. One is the accumulation results in a lower pH compared to cytosolic pH, but it also provides the proton motive force that this protein, VMAT2, or the vesicular monoamine transporter 2, needs to sequester dopamine within the synaptic vesicle. So VMAT2 exchanges these hydrogen ions and then moves dopamine from the cytosol into the synaptic vesicle. So this is the packaging of dopamine that's necessary for synaptic transmission to occur. And what we are interested in is what happens if VMAT2 is unable to package dopamine into synaptic vesicles. Because when that happens, we know that dopamine will accumulate in the cytosol. But when dopamine accumulates in the cytosol, it participates in two metabolic processes that can generate reactive metabolites and reactive oxygen species and cause detriments to the neuronal health. So just to summarize that again, we know that dopamine, through these two cytosolic processes, oxidation and enzymatic degradation, acts as an endogenous neurotoxin because of the reactive metabolites and reactive oxygen species. And so I will just briefly describe both of these processes, the oxidation and enzymatic degradation, starting with dopamine oxidation. So dopamine in the cytosol, based on its chemical structure, it will rapidly undergo either auto-oxidation or enzymatic oxidation, and this process forms the highly reactive dopamine quinone. And whether this is a 1 or 2 electron reaction, it will generate either superoxide or hydrogen peroxide as a byproduct as it is oxidized. And both of these, superoxide and hydrogen peroxide, are reactive oxygen species. But the dopamine quinone is also very detrimental to the cell because it's highly electrophilic and it's vulnerable to nucleophilic attack. And so this is commonly reacting with sulfhydryl groups that are found on free or protein-bound cysteine. And cysteine is commonly found in the cell in the form of glutathione, which is an endogenous antioxidant, but it's also found at the active sites of proteins. So when dopamine interacts with cysteine here, this is a covalent modification, and it can squander the antioxidant defense that would otherwise be mitigating these reactive oxygen species that are being produced and preventing oxidative stress. But when it's binding to the active site on proteins, it can also render them inactive and signal for their degradation. If dopamine does not undergo oxidation, it's susceptible to enzymatic catabolism. And so one pathway by which this occurs is starting with the enzyme MAO, monoamine oxidase, which converts dopamine to this aldehyde metabolite, generating hydrogen peroxide in the process. So again, the production of reactive oxygen species. Dopal can undergo further catabolism by aldehyde dehydrogenase to form dopac. And so in addition to the production of the reactive oxygen species, both dopal and dopac are susceptible to oxidation, just like dopamine would be, and they can interact with other cellular components too. So both through the oxidation or enzymatic catabolism of dopamine, you have all of these reactive metabolites that are going to be interacting with proteins, lipids, everything else in the cell, as well as the production of reactive oxygen species. But normally, these processes are limited because VMAT2 is sequestering dopamine within the synaptic vesicle. And once dopamine's in the synaptic vesicle, it won't undergo these processes. However, there is significant evidence that VMAT2 is dysfunctional in Parkinson's disease. So similarly to what I showed earlier with the pathology of Parkinson's disease, we can look at VMAT2 immunoreactivity in the striatum. And so in a control brain, we can see this dark and dense staining indicative of VMAT2 in the terminals, but in tissue from a Parkinson's patient, there is a loss of this staining. And so this could be just due to the degeneration of the terminals. They're losing VMAT2 because they have degenerated, or it could be that the remaining terminals aren't expressing as much VMAT2. VMAT2 that is isolated from the striatum of Parkinson's disease patients, when put through an uptake assay from both the caudate and the putamen, it's shown to have a decreased activity rate. So this means that not only is there less VMAT2, the VMAT2 that's remaining is less able to sequester dopamine into synaptic vesicles. And this also corresponds with an increase in dopamine metabolism that's been measured in postmortem tissue from Parkinson's patients. Something I find to be the most interesting, though, is that in circulating platelets taken from Parkinson's disease patients, when VMAT2 mRNA is analyzed, there's decreased VMAT2 mRNA in Parkinson's patients compared to healthy controls. So this suggests that there might be a systemic deficiency in VMAT2 that is involved in the disease pathogenesis. The gene that codes for VMAT2, SLC18A2, it has been analyzed to see if there are variants associated with Parkinson's disease. A few have been identified, but predominantly this is found with variants that increase VMAT2 expression, and this is associated with a decrease in Parkinson's disease risk. There is a very rare variant in VMAT2 that is believed to decrease VMAT2 activity that has been identified, and that is related to an increase in the risk of Parkinson's disease risk. But this is a very rare variant, and so it's not believed that any variants in VMAT2 are associated with the development of Parkinson's disease. So just to summarize my introduction thus far, we know that genes and environment are interacting to cause pathogenic mechanisms that result in the development of Parkinson's disease. And we know that Parkinson's disease is characterized by an increase in cytosolic dopamine, or dysregulated dopamine. I did not show this, but we also know that this increase in cytosolic dopamine can interact, it can feed back and interact with these pathogenic mechanisms and further contribute to the disease cycle. But what we were really interested in was what would happen if we started here with an increase in cytosolic dopamine, and if we increase cytosolic dopamine, would we be able to reproduce the features of Parkinson's disease absent of any of these other genetic or environmental factors? So this led to the hypothesis that the packaging of dopamine within synaptic vesicles would modulate the health of dopaminergic neurons. And we were not the first to have this hypothesis. Others before us had created model systems to investigate this question. Here I'm showing some data from a Drosophila or fruit fly model that has a loss of function mutation for VMAT2. And in these fruit flies, there are fewer dopaminergic neurons, but also enhanced neuron loss when the fruit flies are exposed to rotenone. And rotenone is a pesticide that has been linked with the development of Parkinson's disease. In mouse models, we have learned that VMAT knockout mice are non-viable. However, there is this hypomorph model that is expressing only 5% of the normal amounts of VMAT2. And with these mice, they show an age-dependent neurodegeneration, so a loss of the cell bodies in the substantia nigra and the degeneration in the striatal terminals. But if you can see here, this is only starting really occurring at 18 or 24 ages, or months here. So this is aging the mice significantly before you start to see the degeneration. And so our objective was to create a new model that would allow us to manipulate VMAT2 expression in the brain of an adult rat using gene therapy techniques. And this model uses viral-mediated RNA interference. And so right here, I'm just showing plasmid maps of the two viruses that I'll be showing at the beginning here, where these viruses are adeno-associated viruses that express shRNA against VMAT2, or here, this is just a scramble control. And the way that this RNA interference works is that shRNA targets mRNA for VMAT2. And when it's expressed in the neurons, it will prevent the translation of VMAT2 mRNA. So it will block the translation of additional VMAT2 after the point at which the neurons are transduced with this virus. One benefit to this method is that we can target the VMAT2 loss within specific neuronal populations. So in my experiments, I injected this virus targeting the dopamine neurons in the substantia nigra, just directly looking at these neurons affected in Parkinson's disease. This is in contrast to genetic models that would have a global reduction of VMAT2. So this would be experienced everywhere in the body or in the brain. The other benefit to this is we can do unilateral injections. And so we're only affecting half of the brain. And this allows for an internal control in the contralateral hemisphere. We can also do these injections when the rats have reached adulthood. This is also in contrast to the genetic models where these animals would be experiencing a deficiency from the point of conception. We can let the animals age normally and then acquire the loss once they've already reached adulthood. This also allows for a faster time course of experiments. So compared to the mouse model, which took 18 months before you started to see degeneration, in our models, we can look at them in a matter of weeks post-viral transduction. Another benefit is that you can modulate the extent of knockdown. This is by diluting the titer of the virus. So this is changing the number of viral particles that are delivered or modulating this time point. So how many weeks after the viral transduction are you performing the analysis? So right here, this is just a generic outline of the experiments that I'll be presenting, where animals, adult rats, received unilateral injections, either of our SHVMAT2, the VMAT2 knockdown virus, or our control. And then after a post-transduction period, the animals were sacrificed and the brains harvested for tissue analysis. And I think I saw a raised hand if somebody had a question.
You may have answered it already along the way.
Okay,
I
great.
think it's okay to go on.
Okay. So the first thing that we wanted to do in our model was make sure that the virus was actually having the effect that we intended. We wanted to make sure that this virus would decrease VMAT2 expression. And so what we're looking at here is the midbrain of an adult rat. And what we are looking for are the transduced dopamine neurons in the substantia nigra. So to identify dopamine neurons, you can stain for tyrosine hydroxylase. And what I've marked off here with this dotted line is the region of the brain that is the substantia nigra. This region right here, this neighboring region that also expresses TH, they're also dopamine neurons, but this is the ventral tegmental area. And it is seemingly less affected in Parkinson's disease. So we'll be focusing right here on the substantia nigra. And what we can see is that in the dopamine neurons, we see viral transduction as evidenced by GFP. The plasmids express GFP that allow us to identify transduced neurons. And in these neurons, there's a loss of VMAT2 protein compared to the non-transduced hemisphere, which is still expressing VMAT2. And looking at higher magnification, we can see that in these virally transduced, so they're GFP positive, TH positive dopamine neurons, if you inject with our control virus, there's still VMAT2 expression. However, when you inject with our VMAT2 virus, in these transduced neurons, there is a loss of VMAT2 expression. And so in animals receiving our knockdown virus, they express about 50% of the amount of VMAT2 as compared to animals that received the control virus. And this was specific to VMAT2 because we performed this quantification on TH to measure just another dopamine protein to make sure that our effect was specific, and it was. So this is in the cell bodies in the substantia nigra, but looking at the terminals in the striatum, we can look at the immunoreactivity of VMAT2. And in the non-transduced hemisphere, we see, again, this nice dark staining, which is indicative of VMAT2 in the dopamine terminals. But on the transduced hemisphere, there is a loss of this protein expression. So this does appear that there is a loss of VMAT2 protein. And when quantified, we see that this is, again, almost about a 50% loss of VMAT2 in the transduced hemisphere compared to the non-transduced hemisphere. So seeing that there's a loss of VMAT2 protein, what we would expect is that as a result, there would be dopamine accumulating in the cytosol, which we know when dopamine's in the cytosol, it's susceptible to metabolism. So forming things like dopal or dopac, as well as the dopamine quinone. And so what we wanted to do was measure or look for these metabolites here, look for dopamine and look for dopac and dopamine quinone. And so this analysis was performed at 3 time points, 4 weeks, 8 weeks, and 12 weeks post-transduction. First, we're just looking at dopamine. And we can see that starting as early as 4 weeks, there is a decrease in the amount of dopamine in the transduced hemisphere. This is in the striatum, compared to the non-transduced hemisphere. And this is persistent and progressive. You see this decrease progress at later time points. But the dopamine that we'd be measuring here, this is the dopamine that's within synaptic vesicles, because we know when dopamine's in the cytosol, it's very quickly going to participate either in enzymatic degradation or oxidation. So this dopamine that we're measuring is actually the vesicular dopamine. So seeing this decrease over time does suggest that there's a loss of VMAT2, a loss of dopamine being sequestered into synaptic vesicles. So to measure enzymatic degradation, you can look for dopac and create a ratio of the amount of dopac to the amount of dopamine. And as this ratio increases, that would suggest that there's an increase in the amount of degradation occurring. And so we see that at 4 weeks, there's not a significant difference. However, starting at 8 weeks, there is an increase in this ratio. So this does suggest that there's increased enzymatic degradation occurring. The dopamine quinone, as I mentioned, is highly reactive and it's going to bind to cysteine. And so measuring the dopamine quinone itself is not very possible because it is a very transient species. It won't stick around. So instead, what you can do is look for dopamine cysteine adducts. So this is where dopamine has bound to cysteine, either free cysteine or protein bound cysteine. And we do see at the 8-week time point that in the transduced hemisphere, there is an increase in the presence of cysteine dopamine adducts. So both of these analyses here suggest that there is an increase in cytosolic dopamine participating in these two processes that we know allow dopamine to act as an endogenous neurotoxin. And so that's what we really wanted to know. If you increase cytosolic dopamine, is this going to cause the neurons to degenerate? So of course, that's what we looked at next. We wanted to see whether this was actually causing a loss of the dopamine neurons. First, we started looking in the substantia nigra. And to evaluate this, I performed a semi-automated fluorescent method of stereology, where you do a triple stain, first MAP2 here in red. Which is a general neuronal marker. It's going to mark all neurons. DAPI here in blue, which is just a nuclear marker, and then TH in green, which is going to identify the dopamine neurons. And what I did was look for overlap between the three channels here, and where there was overlap between all three, we could count that as a dopamine neuron. And we can count the number of dopamine neurons in the non-transduced hemisphere and compare it to the hemisphere that's been transduced with the VMAT2 knockdown virus. And when this quantification was performed, we saw that there was a loss of about 40% of the number of dopamine neurons in the transduced hemisphere compared to the non-transduced hemisphere, and this was at 6 weeks post-transduction. So we do see that there is a degeneration occurring at the level of the cell bodies. We wanted to see whether this was also occurring in the terminals in the striatum. And so again, looking at TH immunoreactivity, we can see in the non-transduced hemisphere this nice dark staining. However, in the transduced hemisphere, we see this very focal loss or focal lesion of TH in the dorsal lateral striatum. And this is very typical. It's seen in a lot of models of Parkinson's disease because the terminals that are here in the dorsolateral striatum correspond to the cell bodies in the substantia nigra. So this is corresponding to that loss of the cell bodies that we saw in the previous slide. And when quantified, we do see that this is a significant decrease in TH expression in the striatum. So in Parkinson's disease, we know that this dopamine degeneration is causing the motor symptoms. And what we wanted to do was see if our animals were also displaying any deficits in dopamine-mediated behaviors. So I performed two behavioral analyses here. The first one I'm showing is the postural instability test. And in the postural instability test, this is mimicking a test that is done in the clinic where a clinician will take a Parkinson's disease patient and slightly tilt them off balance. And the amount of time or the amount that this patient is moved before they recognize that their center of gravity is off and they take a step to regain that center of gravity or restore their balance is indicative of a postural instability. So as that amount of time increases, that is indicative of postural instability. So to model this with our rats, what we did is we hold a rat with its back legs off of the surface, and we can test each of the forepaws independently. So we'll start with just one forepaw, and the other forepaw will be restrained against its body. And so that one forepaw that is being tested is down on a surface, and an animal is moved horizontally parallel to the surface. And the amount that this animal is moved before it recognizes that it is off balance and needs to take that corrective step is measured. And we call this distance, the distance to trigger. So it's the distance the animal is moved before it is triggering that corrective step. And the benefit of this is that you can test each paw independently. And in a healthy paw, the animal will be moved around 2.5 centimeters before it takes that corrective step. And so what we see in the ipsilateral paw, so this is the paw that is on the same side of the body that received the VMAT2 knockdown virus. The paw is performing at a normal healthy baseline level of 2.5 centimeters. And this is expected because we know that there is contralateral control from the brain to the body. So the contralateral forepaw is actually going to be the forepaw affected by VMAT2 knockdown. And we do see this increase in the distance to trigger. And so this is suggested that the animals are displaying postural instability. The other behavioral test that I performed was the cylinder test. And in this test, rats are placed in a clear cylinder and allowed to freely behave for a 5-minute period while the investigator leaves the room. So the animals are recorded on video. And then the videos can be scored, and the number of times the animals use each of their forepaws as they rear and explore around the cylinder are counted. And what we saw was that the contralateral forepaw, so this is the affected forepaw, is very rarely used during rearing behavior, where they still will use their ipsilateral paw during rearing behavior. So this does suggest that in addition to the postural instability test, through the cylinder test, we are seeing these deficits in dopamine-mediated behaviors. So as just an interim summary, what we've been able to see is that our virus is resulting in a loss of VMAT2 protein. This is happening in the cell bodies in the substantia nigra, as well as in the striatum at the terminals. This loss of VMAT2 protein is resulting in an increase in dopamine metabolism. And this increase in dopamine metabolism, which we know is producing, sorry, I have a cat that's trying to make an appearance. Producing the neurotoxic species is causing nigrostriatal degeneration. And this loss of the cell bodies and loss of the terminals, creating a deficiency in dopamine transmission, is resulting in the behavioral deficits. But what we really wanted to know, now that we've seen that we are mimicking Parkinson's disease, are any of those pathogenic mechanisms of degeneration occurring? So the ones that I mentioned at the beginning: oxidative stress, mitochondrial dysfunction, or protein aggregation. So starting with oxidative stress, we know that the cytosolic metabolism of dopamine is generating reactive oxygen species. And we know that normally reactive oxygen species are kept in check by antioxidants. But with an increase in the production of reactive oxygen species, the antioxidants are no longer able to mitigate this, and you have what's called oxidative stress. And so to measure oxidative stress, what we did was look for oxidative damage in the cells. And to do that, I looked for 2 species. The first one, looking for indices of oxidative stress by looking for oxidative damage. I looked first at 3-nitrotyrosine. This is a protein modification. And we did see that this was significantly increased following VMAT2 knockdown. Just another indice of oxidative damage, 4-hydroxynonenal, when quantified, we saw also resulted in a significant increase. So this does suggest that oxidative stress is occurring, as we can see through the presence of oxidatively damaged proteins. And we know that one thing that oxidative stress can do is induce the activity of a kinase LRRK2. So this is a leucine-rich repeat kinase 2. And gain-of-function mutations in LRRK2 are the most common cause of genetic Parkinson's disease. And this kinase, when it is activated, it will autophosphorylate. So it'll phosphorylate itself as well as members of the Rab protein family. These proteins are involved in intracellular trafficking. So in addition to it phosphorylating itself, it will phosphorylate Rab proteins. And so if we want to measure whether the oxidative stress that we're seeing following VMAT2 knockdown is inducing LRRK2 activity, we can do this through the proximity ligation assay, which will amplify the amount of autophosphorylated LRRK2. And so, as I mentioned, LRRK2, when it is activated, it will phosphorylate itself here at this residue here, 1292. And if we want to measure the amount of LRRK2, we can't just look for using an antibody for LRRK2, the specific phosphorylation, because it's very low abundance, so it's hard to detect. But what we can do is use this assay called the proximity ligation assay. And the way that this assay works, it's typically to identify interactions between 2 neighboring proteins. And so you use a traditional primary antibody against your 2 proteins, But the secondary antibodies that you use have these oligonucleotide tails. And when they're close enough in proximity, they will undergo ligation and cyclic amplification that incorporates a fluorescent signal. So where there is a fluorescent signal, you can identify interaction between these two proteins. But the way that we used this to identify autophosphorylated or activated LRRK2 is we used two antibodies to LRRK2, one of them for the specifically autophosphorylated LRRK2 and another on a different region of the protein. And so using those two primary antibodies and then using the secondary antibodies that will produce a fluorescent signal for activated or autophosphorylated LRRK2, we can look for that fluorescent signal. And what we saw was there was a significant increase in the amount of PLA signal in the transduced neurons. So it does look like downstream of VMAT2 knockdown, there is an increase in the activation of this kinase LRRK2. This is looking at the autophosphorylated LRRK2, but another thing you can look at is the phosphorylation of its substrates. So looking at those Rab proteins that we know that it is regulating. And so I evaluated this by looking at the amount of phosphorylated Rab10, just one of these Rab proteins that we know is a substrate, and we can see that this is increased within the transduced neurons as well. So in addition to measuring an increase in the amount of activated LRRK2, we also see an increase in the amount of phosphorylated substrate. This suggests that there really is an increase in the amount of activation in LRRK2. So changing gears slightly, we also wanted to look at alpha-synuclein. So as I mentioned, synuclein is found in Lewy bodies, and gene variants in the synuclein gene have been associated with Parkinson's disease. These are things like point mutations, gene duplications, or gene triplications. And alpha-synuclein is necessary, it is required for proper neuronal functioning, but aberrant forms, so when synuclein is either from these point mutations becoming modified, or it's having an increase in its expression through the duplication or triplication, it can become aggregated and insoluble. And this is where alpha-synuclein starts to become toxic to the cells. And so the first thing that we looked at was just the total amount of alpha-synuclein. And we did not see any significant differences in the amount of alpha-synuclein following VMAT2 knockdown. This was not entirely surprising, as we know that this alpha-synuclein that we're looking at here, this is just total normal alpha-synuclein. And what we were really interested in was whether there were any changes in the pathogenic forms of alpha-synuclein. And so one of these pathogenic forms is the phosphorylated alpha-synuclein. So this is synuclein that is phosphorylated at this residue 129. And the phosphorylation of synuclein can increase its likelihood to aggregate and form these pathogenic species. And what we saw was within the transduced neurons following VMAT2 knockdown, there was a significant increase in phosphorylated synuclein. And one of the things that aberrant alpha-synuclein can do is cause mitochondrial dysfunction. And one of the ways that this can be measured is, again, using the proximity ligation assay, this time in the traditional method. So looking for interactions between alpha-synuclein and this protein TOM20. TOM20 is a mitochondrial protein. It's found on the mitochondrial membrane. It's involved in protein import. And what we did was look for interactions of synuclein and TOM20. And when this assay was developed, it was shown that with normal, healthy monomeric alpha-synuclein, this interaction does not occur. So it's only occurring with the aberrant forms of alpha-synuclein, the pathogenic forms. This is either an aggregated or modified. Dopamine can modify synuclein, or phosphorylated synuclein, as I showed on the previous slide. And so looking at the proximity ligation assay between alpha-synuclein and TOM20, we can see that within these transduced neurons, there is an increase in the proximity ligation signal. So this does suggest that not only did we see from the increase in phosphorylated alpha-synuclein that there are the presence of pathogenic forms, but the pathogenic forms seem to be interacting with mitochondria. When alpha-synuclein binds to TOM20, it blocks protein import and it causes mitochondrial dysfunction. So to summarize what I've shown so far, we've been able to show that a progressive and acquired loss of VMAT2 in adulthood was sufficient to induce Parkinson's associated pathogenic mechanisms. So we saw that there was decreased VMAT2 protein expression. This resulted in dysregulated dopamine biochemistry, so the increase in dopamine metabolism. This resulted in degeneration of dopamine neurons, and the degeneration of neurons resulted in dopamine-mediated behaviors. And as I was just showing, evidence of these Parkinson's-associated pathogenic mechanisms, so things like the formation of aberrant alpha-synuclein or the induction of LRRK2. And just in the interest of time, I didn't show all of the data, but all of these experiments were also performed in animals that received the control virus and also in female animals that received the knockdown virus. So it did appear that our virus did what we hypothesized that it would do. By decreasing VMAT2, we were able to induce Parkinson's disease, but we wanted to make sure that this was actually a function of a loss of VMAT2. And so to address this, what we wanted to do was rescue this pathology by reintroducing VMAT2. And so for this rescue of VMAT2, this has not shown up correctly, but this should say KDR VMAT2 here for knockdown resistance. What we wanted to do was create a second virus, so our knockdown resistant virus, that could be used with our knockdown virus to restore VMAT2 expression. So what we needed to do was look at the shRNA target that our VMAT2 knockdown virus was targeted against. So it's targeted against this nucleotide sequence. And we looked at the human VMAT2 sequence, and there's actually a single nucleotide difference here. And what we did was introduce more silent mutations. So these silent mutations change the nucleotides, but they do not change the amino acid. So the translation of this sequence here would result still in the production of functional VMAT2, but this nucleotide sequence would not be recognized by the shRNA in our knockdown virus. So we took this sequence and made a second virus, this knockdown resistant virus. And in these experiments, we were comparing if you knocked down VMAT2 and just as the intervention had a control GFP virus, how did that compare to when you knocked down VMAT2 and you restored VMAT2 expression with our knockdown resistant virus? And so just first we saw that by knocking down VMAT2 and just having our GFP control virus, there's still a loss of VMAT2 protein. But when the knockdown resistance virus is also introduced, you see a partial rescue of VMAT2 expression. So it's not quite to these control non-transduced levels, but it still is significantly higher than the amount of VMAT2 just resulting from the knockdown without the intervention. And so this is great. We were hoping to see that our knockdown virus would reintroduce VMAT2, but what we were most interested in was whether this would prevent against the pathology. And so to do that, I performed a stereology again to count the number of dopamine neurons in the substantia nigra. And comparing to the animals that received the knockdown virus with no rescue, the animals that received the rescue virus showed protection against degeneration. So they had a significantly higher number of dopamine neurons in the substantia nigra compared to the animals that received the knockdown virus. So to summarize, we were able to create a novel model of progressive acquired loss of VMAT2 in adulthood, and this is used in rats. Our virus has also been tested in mice. It can also be used in mice. We replicated some of the findings from previous models of deficient VMAT2 expression, but we were the first to demonstrate LRRK2 activation downstream of VMAT2 knockdown. We know that LRRK2 can be activated by oxidative stress, but it's never been shown that this can occur downstream of dopamine dysregulation. So we provided some further evidence that suggests dopamine may be involved in the pathogenesis of Parkinson's disease. And this does suggest that dopamine homeostasis may be a therapeutic target for Parkinson's disease. So I mentioned at the very beginning of my talk, we have symptomatic relief for Parkinson's disease, and this is commonly in the form of L-DOPA. L-DOPA is the precursor to dopamine, and we can supplement with L-DOPA to restore motor function. But what this is doing is increasing the amount of dopamine in the brain. And if we think that there's deficient VMAT2, either through a decrease of protein or a deficiency in its activity, you might be increasing dopamine, but not having the ability to properly sequester it. So you might be having a further increase in cytosolic dopamine, which could be further toxic to the dopamine neurons. And so that concludes my data talk. I'd like to acknowledge my previous mentor, Dr. Teresa Hastings, as well as other members of the Hastings lab, also our partner lab, the Greenemeyer lab, and other members of our Institute's gene therapy group, as well as some funding sources that supported this work. And with that, I'll take any questions.
Wonderful, thank you very much. Again, Zoom clap. That was fantastic, really great. Thank you.
Yeah.
All right, questions, chat, Q&A, hand raised from anybody?
Am I unmuted now?
You are, Janet.
Oh, thank you, Megan. That was wonderful. Great presentation. I don't think my camera's on, is it?
It is not.
Okay.
I don't
I
think.
meant it to be, but I don't think it is. So yeah, that was fantastic. What a great presentation. Congratulations on so much great work. Very nice. So just stepping back to see the forest for the trees, because I don't really know that much about the trees in this particular instance, but to see the forest a little bit, what do you think is going on in terms of dopamine release in your VMAT knockdown? Because as I'm sure you're aware, one of the kind of persistent hypotheses regarding why you don't see an onset of symptoms in Parkinson's patients until the degree has progressed is that, in fact, the remaining dopamine neurons are able to pick up the slack, and so you have to lose about 90% of them before you start to see the motor deficits. And so how do you think that would work with the notion that VMAT is not working properly?
Sure. So yeah, that's an excellent point that I did not mention, that Parkinson's disease diagnoses typically occur once there is about an 80 or 90% loss of dopamine transmission in the striatum. It takes an extensive loss of the neurons before the motor symptoms start to occur. And it is thought that either the remaining neurons or actually also neurons from other areas of the brain can sprout into where the terminals that have degenerated were. And so this can also come from other neurons, such as serotonergic neurons, where they're filling in those gaps and can kind of be helping to prevent against the loss of dopamine causing motor deficits. In my model, we did not actually measure dopamine release through something like cyclic voltammetry. That's something that would have been very interesting. Just no one in my group or at our university had any experience in cyclic voltammetry. But I would suspect that you would see a decrease in at least action potential evoked neuronal transmission of VMAT2. So we know that when there is an increase in VMAT2 in the cytosol, that the dopamine transporter on the plasma membrane, it can reverse and pump that dopamine out into the synapse. So if any of that dopamine in the cytosol that is remaining as dopamine and not being oxidized or metabolized, it could be pumped out through DAT into the synapse, but we would expect that the vesicles, while they might still exist, they're not actually filling with dopamine. So when an action potential comes and they fuse, that there's less dopamine being released.
Right,
Thank you.
Jeff Carroll, go ahead.
Megan, hi, it's Jeff Carroll. Beautiful talk. Your behavioral data was particularly really tight, and as having done a lot of behavior, I was very impressed. So beautiful job on carefully handling your rats there. I just had a question, and I might have misunderstood. I was just trying to keep track of how sensitive cells in vivo are to VMAT2 loss. So in the hypomorphic mice, is it correct that it took quite a while for the cells to die, whereas you saw pretty quick atrophy in your AAV mice? And I wonder if you have thoughts about if that's a discrepancy, and if so, what your thoughts are on why it might arise from.
Definitely. Yeah, no, you're exactly right. So those hypomorph mice, which express 5% of the normal amounts of VMAT2, it takes up to starting at around 18 months before you start to see significant neurodegeneration starting in the substantia nigra. And so what we believe is happening here is, because this is a genetic model, that these mice are probably developing with compensatory mechanisms to help against that loss of VMAT2. So through other mechanisms, this could be through changing the amount of DAT expression, so that maybe the little bit of dopamine that are getting released stays in the synapse for longer, or other compensatory mechanisms, maybe by increasing the amount of dopamine synthesis, that it's protecting against this loss until much, much later in life. And so one of the things that we also thought with our model is that young animals tend to be pretty resilient. And so if we've seen in other models of Parkinson's disease, so like some of our pesticide models, if you try to model Parkinson's disease in young rats, it's highly variable. They are pretty resilient. So a lot of experiments are performed in adult, even retired breeder rats. And because Parkinson's is a disease of aging, and we know it is occurring late in life, we think that there are processes happening with the aging process that are contributing to this neurodegeneration. So we think our hypothesis is, in our animals, that because we used adult animals and we introduced the knockdown without them having the ability to develop any compensatory mechanisms, that they are seeing a greater effect of that knockdown than, say, the hypomorphs might be experiencing. Does that make sense?
Yeah, super interesting potential age-related things to experiment with in future. That's beautiful work. Congrats.
And Megan, we have a few questions in the question and answer box. I don't know if you can see that there, but I can read them out if you'd like me to.
Might be able to. Okay, yes, I can. So from Haley, is there a way to target removal of cytosolic dopamine to control Parkinson's symptoms? This is a very interesting question. So the only way that our bodies can naturally target this cytosolic dopamine would be through either the sequestration or the degradation of dopamine. It's possible that somebody may be able to synthesize something that, like a protein, that would bind to and sequester that cytosolic dopamine and pull it out of solution so it can't render any damage. But in terms of endogenous mechanisms, the only way that that cytosolic dopamine is modulated is through the sequestration or the degradation. And then Spencer asks, are you able to see Lewy bodies within the dopaminergic neurons themselves? And what other areas of the brain can Lewy bodies be found, and what is their effect? So Lewy bodies are found throughout the brain in Parkinson's disease, and it's very controversial whether Lewy bodies are detrimental or actually protective. So Lewy bodies are these aggregates of the pathogenic and insoluble proteins. And so one hypothesis is that Lewy bodies are taking all of these dangerous, potentially dangerous molecules and sequestering them into a Lewy body, which then cannot interact with other components of the cell. Others believe that the Lewy bodies themselves may be causing some of the damage. So we did not look for Lewy bodies per se in this analysis. We did look for the presence of aggregated alpha-synuclein, so that would be suggestive of the formation of Lewy bodies, because we know that alpha-synuclein aggregates in Lewy bodies. We did see some pockets, some areas of aggregated alpha-synuclein, but in terms of actually identifying it as a Lewy body, we did not do the analysis to distinguish that from just aggregated alpha-synuclein. There's a question from Jeff here saying, curious if there is a VMAT2 potentiator, so a positive allosteric modulator, available for research. This is a great question, and it is a focus of my current research, trying to identify VMAT2 PAMs, positive allosteric modulators, or other compounds that may enhance VMAT2 activity, because Absolutely. If we had a drug that can increase VMAT2 activity, that's something that we could give to Parkinson's patients alongside L-DOPA, so that when they're producing more dopamine, that dopamine is more able to be sequestered into synaptic vesicles. There is one drug that I know of that has been identified as a enhancer of VMAT2 activity, and the name is escaping me now, but it is actually an FDA-approved cancer drug that was detected in a screen on those deficient Drosophila that I showed some of the data from. So this work was done by Hakeem Lal when he was at UCSD, and he did a screen on those deficient Drosophila that showed motor symptoms and looked at whether any FDA-approved drugs in their library could rescue those motor symptoms, and they did identify one, but we would like to find more.
Wonderful. Well, it looks like there may be no other questions, but let me echo the sentiment of everyone else so far and congratulate you on a really fantastic body of work and on a wonderful presentation. So that was really great, and we really appreciate you being here. And again, I don't know how to clap, group clap on Zoom, but we can Zoom clap, golf clap. Really great. Thank you so much, and thanks to everyone who came out tonight and attended the talk as well.
Yeah, thank you for the opportunity to present.
Pleasure. Thanks, Megan. And thanks, everybody. And thank you, Andrea.
Oh, yeah. Thank you, Megan. It was a
That
great
went
talk.
very smoothly. And thank you, Robert.
You're welcome.
Yeah,
I
it
wrote
was
a
very
message
smooth.
to I wrote a message to Andrea, but I'll just say it out loud. We can edit the presentation with the corrected slides if you're interested.
Oh, really? Okay. Would that just involve me sending you the slides? And okay, yeah, I can do that.
Yeah, you would export them as JPEGs,
Okay.
and then we could run this through my student workers and basically go through and insert the new slide for the old slide. You wouldn't see the little red dot though. You would lose that,
Okay.
but it would be more inclusive of your research and findings.
Yeah, absolutely. That would be wonderful. I can export those slides as JPEGs and get them over.
And
yeah, thanks, Robert.
You're welcome. And as far as the problem, it may be that your computer doesn't have a lot of horsepower or something is clogging it, because I've seen that sort of thing happen before, and it's usually that's the cause.
Okay, yeah, that's good to know. I have been having some issues with this pretty old computer that was with me through all of grad school, so maybe time to replace it.
But just so you know, Megan, I mean, I think it would be great to get the updated slides on the video, but just so you know, it didn't have any impact on your presentation. Everything was perfectly clear, and what you were saying and presenting and the logic you were following was perfectly obvious. So yeah, don't feel bad about it. It was still fantastic.
All right.
Yeah. All right. Well, I'm going to take off. Wonderful. Thank you all again, and good luck. Megan, good luck with your studies. It looks like you're headed in a great direction.
Thank you so much.
Yeah, take care.
All right. Is there anything else you need from me, Andrea? Or are we all good to go?